Yes, the interesting thing is that the SPMs have always been studied in many tissues but the brain. So there's not much knowledge about the brain, and so we are one of the few groups in the world that we are studying these in the brain. And what I can tell you is that so far there are only few evidence on the role of SPMs on specific brain areas, but all these studies have been performed on immortalized cell lines, so models of these, and very few studies on actual human patients. So we are trying to do this in my lab and to study directly these involvement of SPMs in patients. And the role that they play is that they regulate many aspects, because we have evidence now that they can be produced or be taken up by different brain cells, from neurons to glial cells, and mainly infiltrated immune cells that from the blood go inside the brain. So that's for the Alzheimer's disease, that's the most studied ever with the SPMs field, because first of all, because it's the most common neurodegenerative diseases. So it's more easily accessible the human patients, and many studies have been performed by also Charles Saran group, and so we know that the SPMs work on different cells of the brain, specifically on microglial cells. So the microglia, they can uptake the SPMs, and their role is to modulate the conversion from pro-inflammatory to pro-resolving microglia, and also they can enhance the removal of the debris, including the beta amyloid. So that's what has been known so far about Alzheimer's disease. This is actually the main topic that I've been working in the last 5 years, meaning that we are trying to prove the point that the brain is not an immunoprivileged site anymore, like it's always been thought in the last 100 years. So the actual immune cells go inside the brain, and they try to either protect from inflammation going on in the brain and also resolve it. So this was like dogma that for century was not thought of. So because the blood-brain barrier is always thought to protect the brain, so no immune cells can have access to the brain. This is not true, which makes sense, because how can you resolve an inflammation in the brain if it happens in the brain if you don't have immune cells going on there? So the microglia and astrocytes is not enough to do that. You need leukocytes, so you need monocytes, macrophages, T cells to go in the brain. And so this is a new field for resolution, because the role that SPMs do that are according to this recruitment in the brain. And so we are studying all this process of entering in and out of the immune cells, and whether they are able then, once they arrive in the brain, to produce SPMs, or if they have a defect in the SPMs production. So this is what's going on in the last 5 years in my lab, and in some other very few labs which we are trying to study this. In terms of therapeutic potential, the first thing is to actually determine if a patient has a defect in producing SPMs or in responding to SPMs. So the therapeutic potential will be according to these results. So we need to first analyze the patient, and this is specific to the different neurological disorder. So people with Parkinson or with MS or with even headache, they might have a different profile of SPMs, and we already observed that in MS patients and Parkinson's disease, you have different resolvings that are distinct or distinctly modulated according to the disease. And once we know that, then we can, in the future, provide with a therapeutic target using that defective SPM for that specific disease, and even better for a specific person, because the ultimate goal is to do like a personalized, of course, medicine by delivering directly that SPM that's been defective in that specific person, because especially when you talk about these neurological disorders, you're not talking about something which is acute, but it's chronic. So those people have maybe defects which going on for decades, and that's when you might have different profile according to the stratification of each phase of that disease. For every patient, we get the blood and the CSF. So this also is important because we have an analysis of a profile of what happens in the blood and what happens in the brain. And because these two tissues also communicate with each other, we might also understand what's going on in the inflammation resolution, either in the brain or in the blood. So that's the starting point. We need to collect those tissues from the patients, and sometimes it's not easy because for specific diseases like MS, it's very easy because the collection of blood and the CSF is part of the clinical routine, because it's a diagnostic tool. For other diseases, it's not, so that would be more difficult to get the CSF, for example. But I think it's very important to have both, because if you find a defect in the blood, doesn't mean that then the very same cell will be in the brain in that very same patient. So we need to also have to observe what's happening in the brain, and because you cannot get brain tissues directly from patients unless they have a specific brain surgery for other reasons or after they die, then we have to get the CSF. It's the only tissue that reflects what's going on in the brain in terms of inflammation or resolution. Once we get it, we can easily run the analysis of the SPMs, and also in the cells, because from the blood but also in the CSF you get the immune cells, and then we analyze each immune cell subset if they bear any deficiency in the enzymes or in the receptors, so we can also understand whether they are able to produce or to respond to them. The biomarkers are the same in terms of analysis because we analyze everything, so the whole lipidome of pro-inflammatory lipid mediators and pro-resolving lipid mediators, but at the same time we also measure all the genes and all the proteins that are involved in their metabolism and in their function. So all together, then we can really tell where is the defect, or if there is a defect, and in which subtype of patients, and according to the disease phase as well, because also it's important to collect the patient's tissues from different stages of disease, because then we can also correlate with the activation state, the progression phase, and then tell, okay, that lipid mediator is correlates with that onset of disease or the progression of disease, and so on. We have, because I work on MS and Parkinsons disease, so the most patient that I get in touch with is those diseases in the lab, in the clinics that we work in. So people with MS and Parkinson's, they've already been taking this under advice, of course, so, along with their own disease-modifying treatments, of course. They're not using any other, it's not a substitute, of course, so along with their own treatments. So many of these people are already taking the SPM active from Metagenics, of course, and they see some benefits. And what they do is that they take some pills when they have some symptoms, so only a few of them I can see that they can take every day because they are costly, of course. So what I suggest them in that case, just take them when you have a specific symptoms, which means maybe an acute phase or relapse, and they see that they have benefits in that. And according to this, then we also will do a clinical trial on with these products on MS patients, which is under approval, and we will be able to enroll 60 patients with MS, with relapsing remitting MS, and giving them 2 pills of the SPM active products twice a day. And we'll see the results after 2 months and 3 months. So the end point will be 2 months and 3 months, and see what happens in terms of amelioration of the relapse, and in terms of course, production of SPMs in the blood, because I will also analyze that, after 2 months and 3 months, and then also how the immune system is suppressed by these SPMs. So I will analyze all the T cells, which are the main players in MS, which will go in the brain and destroy the myelin. So I will also analyze all the immune cells, but this is still under approval, and we hope to begin very soon this year. But we are already doing, let's say, because our findings are done in the human, so it's already translated into clinical, which means that at this stage we already know what is the defect that the patient has. So the next goal would be just to use this information to make some drug delivery products that are able to restore this defect, and there are some possibilities. That's the hardest part that we've been trying for many years, but also Charlie the same, because that's the most difficult and tricky thing to do, because we need to actually find a way to produce high amounts of this molecule in a non-costly way. That's one of the most difficult thing to do. We need lots of money to do that, and also find a way to actually be sure that this resolving will go into the right direction. Because let's say if I see that the defect is in a specific brain area, and that's what we already know from some of these diseases that I'm studying, so MS and Parkinsons, how make sure that this molecule that I deliver will actually go to that area and not to another area, because that will make a big difference, because I want only to target a specific area where I've seen that there is the defect, So we need to construct specific carriers, the nanoparticles that we are doing now. It will take many years to do that, but this is the ultimate goal, so that we can deliver specifically to the area or to the cell that we want the SPM to go, Yeah, the challenges are many, even if the knowledge about them is increasingly, it's increasing in the last 10 years, the challenges are still the same as the one that we had like even 20 years ago. So the first challenge is, of course, the ability to make a compound of higher levels. Technically, it's very difficult to have this drug, which is GMP free, and the production of the drug is very complicated, so we need to find a way to make it easier and less costly, of course. And then the second challenge is, as I was telling before, is that we need to know where this molecule needs to be oriented, so to go. So the challenge is that how can we build a drug that allows us the SPM to go where we want to, because from the moment it enters the body, it will surely be degraded, metabolized, and so that we don't see the effect. It's easy to do this in experimental animal models, where you can inject high dosage or as many times as you want, but you can't do that in human patients. So you need to find a stable way to avoid the metabolism of these SPMs, and that goes in the right direction. So these are, I guess, the main challenges, and also to decide which SPM. And this comes back to what I was telling before, that we need to profile them for each disease and for each phase of disease, and even for each individual, so that we can decide which is the best SPM to give for that patient at that phase of the disease, because it might change, and I'm sure it's different. It's different because we've already seen they differ from different diseases according to the disease phase. So in order to make a product, a drug that contains one of the SPMs, you need to know which SPM to include in that drug. But before doing that, you need to know which patient has that effect of which molecule, because there are so many, because there are more than 30 SPMs, and you need to know which one to choose.